emotions of emotion and stress. Wait, are these aren't the same slides as the one that's on the group meeting. I mean on the drive, on the Google Drive.
Yeah, I didn't make a PowerPoint for emotion and stress, but I did for memory and learning.
I see one for emotion and stress.
I didn't fill that one out though.
Oh, okay, I see what you mean. Yeah, I see.
Is that okay?
No, that's fine. It's fine.
Okay.
I'll just like, I guess just go off the slides and then add to any. I mean, if you want to do this one slide.
Sure. So Phineas Gage was a patient who had a metal rod kind of go through his prefrontal cortex. And because it damaged that area of his brain, it damaged his ability to inhibit his emotions. So his behavior after the accident was incredibly different from his behavior before. He was unable to control most of his emotions, and he had very bad impulse control.
Okay, so there are different theories of emotions. Two of the big keystone theories that we have established is James Lang versus Ken Bard. Two of these basically they're both defying the definition of how we process emotion. However, one of them sees it as we have the stimulus, then we have the physiological reaction, and then it leads to emotional it leads us to defining the emotion. Versus Cannon-Bard, who has the physical reaction. We get the stimulus, which gives us the emotion reaction, which leads to the physical reaction. So they're just arguing which order that the definition of emotion happens. Anything y'all want to add?
No, today they think it's a very complex interaction of both things happening at the same time, and it's not necessarily one or the other.
Mm-hmm.
Yep.
So, like we talked about in James Lang's theory, conscious feelings follow body changes. So, an event in the environment triggers behavioral, autonomic, and endocrine responses. Uh, Feedback from these responses produces emotional feelings of emotion. You go ahead.
Oh, sorry.
I'd say like some evidence towards this would be like arranging the facial muscles to mimic an emotion that can invoke the feelings of that emotion.
Okay.
This emotions as response pattern. So we have three different types. We have the behavioral, autonomic, and hormonal response. In the behavioral, it's appropriate of muscle movements such as like aggressive posture, growl, and showing teeth. Autonomic would be activation of the autonomic nervous system, activation of sympathetic branch increases the fight or flight mechanism, while parasympathetic branch increases heart rate, regulates blood flow towards the muscles. This would be like that rest and digest you guys discussed earlier in this semester. And then you have your hormonal responses, which facilitates activity of the autonomic nervous system. It's a release of adrenaline and noradrenaline, which increases blood flow, converts nutrients to glucose. This example would tie into that flight or flight mechanism. So sympathetic division of the autonomic nervous system and emotions. So just for the autonomic nervous system, just remember the four Fs, which were sexual reproduction, fear. What was the other, do you guys remember? What was the other two? It was
and it was fight and flight.
Fight and flight. Yes. Associate. So like I said, that fight or flight is associated with fear or just arousal, which leads to your pupils dilating, your heart rate increasing. But it's not just that fear that's the only emotion that's there. It could also occur from anger, sadness, like digestive, such as like hunger, or you're surprised, or even like happy. The peripheral components of emotion prepare the body for action and communicate our emotional states to other people. So unless it's like extreme, arousal enhances intellectual and physical performances. That's why when you're in this like a serious, dangerous situation, you might be able to do tasks that you didn't think were like physically possible. You know how they say like a mother will lift a car to get her baby from underneath it? It would come from that
Mhm.
basis. So the Ken and Brad theory, how it adjusts to the James Lang theory, would instead of emotional expression results from the function of the hypothalamus and emotional feeling, and emotional feeling affect the results of the stimulus of the dorsal thalamus. The physiological changes in subjective feeling of emotion in response to a stimulus are separate and independent. Arousal does not have to occur before the emotion. So that's like I said, they just argue over which order that process of emotion happens.
And they used a series of like evidences to prove their views versus the other person's. And some of these included that if you completely separated the organs from the central nervous system, it doesn't like change your emotional perception or your emotional behavior. Cannon-Bard also argued that the viscera are relatively insensitive structures and that they themselves and their responses. Oh, sorry. Can you guys see the PowerPoint?
If you can't, can you try clicking on Divya's screen so you can make that the main screen, perhaps?
Can everyone else see it? Okay. Marin, can you see it now? Mm. uh,
Maybe it's on their end. Try leaving the session and coming back. Right, should we continue?
okay. Yeah.
All right. So this is also part, Ken and Bertrand. They defined anger and what causes it to inhibit. There's something they define as the sham rage, which is undirected biting, clawing, hissing, arcing of the back, and violent limbic movement, produced in experimental animals by removing the cerebellar cortex. The cerebellar cortex is what inhibits those kind of physical anger emotion, physiological anger responses. Uninhibited hypothalamic discharge. 3 known cases in humans would be hypothalamic lesions, carbon monoxide poisoning, insulin, hypoglycemia. So this just shows how the pons and cortex work with one another, and these are some of the causes that would create that. So the hypothalamus coordinates the peripheral expression of emotional state. So this is what basically decides how your body wants to react to such emotional responses.
It just integrates sensory inputs from the outside and contextual information that your cortex will synthesize. And will synthesize all that information and turn that into a physical response.
Remember, honey, can you see the PowerPoint now? All right, let's keep going.
All right. Oh, oh. Okay.
Yeah, so now we're going to go over stress. Um, the HPA axis regulates hormonal stress responses, which this consists of the hypothalamic pituitary and adrenaline. Um, Immune suppression would cause like increased heart rate, increased blood pressure, and increased catabolism, which is digestion. For the limbic system, this is this is the order of the limbic system. You have your hippocampal fibers, which project to your mammillary bodies, which in turn project through the mammaliothalamic tract to the anterior thalamic nucleus. The anterior nucleus then projects to the cingulate gyrus, uh, and then the axon of the cingulate gyrus then project back to the hippocampal formation. Right. I guess this is just important pathways you guys should know for the limbic system.
These are just areas surrounding the limbic system that are also involved with emotional perception and processing.
Right.
Sorry, I think there's a lag.
That's
So we kind of talked about that.
One of the conditions that can affect that would show a compromised limbic system would be Iver-Bucy syndrome. It's a syndrome resulting from bilateral lesions of the amygdala. Amygdala. Chance of finding during investigation of the locus of action of mescaline. Example that'd be like chewing behavior in monkeys. This was just one of the cases. Some of the symptoms that would show in this kind of syndrome would be hypersexuality, visual agnesia, hyperorality, dietary changes, or hyperphagemia, and decreased emotional reactions. So for the amygdala, it plays an important role in mediating emotional behavior and motivation. So evaluates the emotional value of sensory stimulus. It kind of represents like an interface between the limbic system and the extrapyramidal motor system. Just makes that decision on what to prioritize and how to feel.
Yep. Continue. So there's a lot of different brain regions that receive input from the amygdala. You could see them over here on the right. And this is a diagram of the nuclei in the amygdala. And there is a long route and a short route for processing through the amygdala. The short route just goes through the sensory thalamus and through the central nucleus and projects to the hypothalamus, the midbrain, pons, and medulla. And that is for some really quick kind of processing, quick, reflexive, immediate processing that needs to be done in a very short amount of time. And the long route goes through the sensory cortex, so like the parietal cortex. And it goes through different nuclei, like the lateral nuclei, the basal nuclei, the accessory basal nuclei. And that just goes to show that there is more processing involved with the higher order cortex and more of the amygdala. So you'd have a better processed emotion or a better processed decision with this path. And it would be more of like a slower, complicated scenario rather than a really quick, instinctive scenario that is represented by the short route. So when we go back to the HPA axis, hypothalamus, the amygdala, and the adrenaline, the hypothalamus, the pituitary gland, and the adrenal gland work together to produce these emotional responses to stress. And the amygdala plays a big role in producing the fear response. So when the amygdala is stimulated and the emotion of fear is perceived in the brain and your autonomic nervous system, it causes adrenaline to be released into the body. And adrenaline will cause these effects that we see over here. Your pupils will dilate, your hair will stand on end, you will start breathing faster, your blood pressure will increase, and these will all prepare you for like that flight or fight response that you need in really immediate situations. And here's just a diagram showing that what will happen if you didn't have an amygdala. Here, an animal would exhibit fear at the sight of a snake, and if you didn't have an amygdala, you would not exhibit fear at the sight of a snake because you couldn't process it. The amygdala is involved in fear conditioning. Fear conditioning is kind of a process that people use with research on different things like PTSD or tinnitus. What they do is they play a really loud sound. They put a rat in a box and they play like a really loud sound while also putting a rat on this kind of shock grid. And whenever they play the sound, they would also shock the rat. And that would kind of condition the rat to think that the sound is going to hurt them. And that kind of pathway of associating the sound with the shock is called fear conditioning, and it heavily involves the amygdala. The amygdala is also involved with associative learning in the same way. It's in your emotional responses will enhance your ability to remember things and to learn them. And that's associative learning. And your orbitofrontal cortex is this area of the brain, and it is involved with inhibition over the amygdala, and it'll allow you to control your experience of the emotion and the expression of the emotion that's happening. So without the orbitofrontal cortex, like we see in Phineas Gage, we'll see a very uncontrolled, uninhibited expression of emotion, and it's a very different behavior that you see. So it's really important that we have this. And the main role of the orbitofrontal cortex is to inhibit the amygdala. And this is just a really interesting situation we see with psychopaths who don't have emotional conditioning training. So early in your life, you learn to incorporate your emotional responses into your decision making. And if you don't do that properly, then later in life, you're not going to incorporate that in the way you make your decisions, and you won't take into consideration, or you won't have that kind of empathy that allows you to determine whether something is right or wrong or whether you should do it or not. And it shows that in psychopaths, you don't have as much activation of that area. So it's like a physical response. It is a physical circuit that is conditioned when you're young. And this is just kind of a summary that we see here. Sensory inputs go through the thalamus, and these inputs are always processed by the brain in different ways. A really quick kind of circuit in the lower brain that responds to it is the amygdala hippocampus, like HPA axis, through the brainstem, through the hypothalamus, right here. And a higher order processing of this goes through the sensory cortex and the association cortices, for a longer, higher order processing of emotions. So you have it both ways. And that is emotion and stress. And we're going to go over memory and learning as well. Let me pull up that PowerPoint. Okay. Sorry guys, there's a bit of a lag. Let me just wait for this to pull up and then we'll start. Okay, let's get started with memory and learning. So first, we're going to go over some basic definitions. So learning is the process of acquiring new information. Memory is your ability to store and retrieve that information. Amnesia is a severe memory impairment. Retrograde amnesia is loss of memories formed before the onset of the amnesia, so before whatever happened to make you lose your memory. And the anterograde amnesia is loss of memories formed after the onset of the event. So you'd remember everything up to that point, but not after. So these are the stages of memory. First, you'd have your sensory input, and you would encode that information into your short-term memory. Consolidation is what happens when memories move from short-term to long-term. And retrieval is when you retrieve information from your long-term memory to use it. Okay, so there's different types of memory. Declarative memory are kind of information that you need to recall. So there's semantic memory, which is generalized declarative knowledge. So an example of that would be like, what are the days of the week? Episodic memory is detailed autobiographical memory. So that would be, what did you specifically do last week? And you recalling all the events would be episodic. Non-declarative memory are like skills that you learn, like learning how to ride a bike or write or do your hair. Those are those are skills. You can't really say them out loud. So these are like sensory motor, perceptual, cognitive skills that you learn. So the main difference would be like declarative or things that you would like recall and say out loud. And non-declarative are like skills that you need to learn. Patient HM is a patient who had epilepsy. And to treat this epilepsy, he had his amygdala, hippocampus, and parts of the temporal cortex removed. And because of this surgery, he developed anterograde amnesia. So that means that he could not remember anything after the surgery. He remembered his entire life up until the surgery. And then after it happened, he couldn't form any new memories. The hippocampus removal will usually result in memory impairment because the hippocampus is really involved in memory formation. Patient HM's non-declarative memory remained intact, however. He improved and learned tasks, but he never remembered that he did them. He just knew how to do them. Okay, so there's different types of learning. Non-associative learning is learning that all involves only one stimulus. So if you were learning like the definition of a word, that would be like non-associative learning because you're not associating it with anything else. Habituation is a decrease in response to a repeated stimulus. So just getting used to something, getting used to a smell in a room, something like that. Spatial learning is also another type of learning that occurs in your brain. The hippocampus has special place cells that are responsible for spatial learning. Associative learning is learning that involves the association between two stimuli. There's different types of associative learning. So classical conditioning is what you think of when you think of Pavlov's dogs. It's when a neutral stimulus, when it's repeatedly paired with a stimulus that would make you respond, will then make you respond to both stimuli. So with the example of Pavlov's dogs, what he did was he presented food to the dogs as an unconditioned stimulus because the dogs would naturally respond to the food by salivating. And then what he started to do was he started to ring a bell every single time he presented food to the dog. And then as he did that, the dogs began to associate the ringing of the bell with the food. So eventually he just took away the food and just started ringing the bell. And he noticed that the dogs would salivate to the bell, just like they would salivate to the food. And that is an example of classical conditioning. Operant conditioning is an association made between behavior and the consequences of a behavior. So that would be things that involved punishment and reward, like taking away your child's TV privileges every single time they don't do their homework is an example of operant conditioning, because eventually your kid is going to learn to just do their homework if they don't want their TV privileges revoked. Mm. Okay. This is a basic summary of the different kinds of memory. So, like we talked about, declarative memory are things that you can repeat to other people. So, episodic memory is personal details that are relevant to you. Semantic memory is just general knowledge that you would know. Non declarative memory are skills that you need to learn. Cognitive skills like conditioning or priming, or motor skills like riding a bike. Okay. So now we're going to talk about short-term versus long-term memory. Short-term memory is memory that usually lasts about 30 seconds and throughout rehearsal, so whenever you're actually using that information. Long-term memory will last for days, weeks, months, or years, so much longer than short-term. And both of these memories are used in the process of forming memories, which involves encoding, consolidation, and retrieval. Encoding is when sensory information is turned into short-term memory. Consolidation is when the information in your short-term memory is consolidated into long-term memory. And retrieval is when you retrieve information from your long-term memory to use it right now. So like if you were in an exam and you were retrieving information that you had studied beforehand. And this is kind of the process of how memories are formed and retrieved. So all your sensory information from your senses are going to be registered by your brain, and then they're going to be turned into short-term memory. So that's encoding. And if that short-term memory is important and your brain decides that it needs to remember it, then the process of that becoming long-term memory is consolidation. And if you need to use that information in the future, the retrieval of that information is retrieval. So you're just going to get that information back and use it when you need to. So the way memories form in your brain involve physiological and structural changes. Physiological changes at the synapse involve increasing neurotransmitter release at the membranes. So you just have more neurotransmitters there. Um, the process of increasing neurotransmitter release or decreasing neurotransmitter release is what kind of shapes the circuits in your brain. So when you first start forming memories, the circuit between neurons that encodes that memory is going to be pretty weak. If you continue to use that, and if you continue to use that memory, then the pre and postsynaptic membranes that encode those memories are going to be more active, and it's going to strengthen that synapse the more you use it. On the other hand, if you, let's say you remember that information, your brain decides it's not important to remember and you don't need it, then you would see that there's less activity at the postsynaptic membrane and the synapse is weakened, and it would eventually not be consolidated into a memory. So through this process of increasing or decreasing neurotransmitters at different synapses, you can form different memories and shape your brain the way you need it to be shaped. Structural changes at the synapse involved increasing and decreasing the neurotransmitter receptors themselves that are present, and increasing or decreasing or increasing the area of the dendritic spines that are present. They're physical changes that you can see. Okay, so long-term potentiation is this process that describes the formation of long-term memories. So how this process starts is it starts with this thing called tetanus. Tetanus is what we refer to when we have prolonged excitation of neurons. So if you are, for example, if you're studying for a test and you're trying to remember the definition of neuron and you keep reading it again and again and again, then the specific neurons in your brain that are encoding that one piece of information are going to be excited repeatedly. And that repeated excitation would lead to tetanus, basically. And after this tetanus has occurred, the excitatory postsynaptic potentials will remain high. So that circuit of your brain is going to be easily excitable. And when that circuit is easily excitable, it's going to produce this process of long-term potentiation. Because the more you remember that one piece of information, the more that one circuit in your brain is excited, the more those synapses are going to be modified, and the easier it's going to be for you to remember that information in the future. It makes your memory stronger because, like we talked about over here, repeatedly exciting those circuits in your brain are going to cause physiological and structural changes. And those changes will make it easier for you to remember that piece of information. And that's what makes some memories stronger and some memories weaker. The hippocampus, whoa, let's go back. The hippocampus is the most important structure for this because that's where it happens, long-term potentiation. Okay, so we're going to go through the process of long-term potentiation. So in your hippocampus, you have specific neurons that encode memories. And in these neurons, you have a lot. So between these neurons, you've got synapses that will look like this. And they've got AMPA and NMDA receptors, which are both glutamate receptors. So when you are trying to form a memory, your neurons are going to be excited and they're going to release glutamate because that's what happens when your CNS is excited. And first, the glutamate is going to activate the AMPA receptor. And the AMPA receptor is going to let in sodium and slightly depolarize, it's going to slightly depolarize the cell. Your NMDA receptor is not going to respond immediately because it's got this magnesium ion in the channel that's going to block it. NMDA receptors with magnesium ion blocks are voltage gated, so they're only going to open up when this postsynaptic neuron reaches a certain voltage. So until it's reached that kind of voltage, it's not going to open this up. So until then, we're just going to stimulate the AMPA receptors. And once enough AMPA receptors have been stimulated, the NMDA receptors will remove their magnesium block and let in calcium. And that's what we see here. And calcium is pretty important because calcium is a secondary messenger that will activate different protein kinases in the cell that will produce different responses. One of these responses is it's going to allow existing AMPA receptors that were in the cell but not at the synapse to go to the synapse and increase the amount of AMPA receptors that are at the synapse. It's going to increase the conductance of ions and increase the conductance of ions because the voltage is going to be higher. And it's going to cause the cell to produce more receptors. So all of these actions are going to make this postsynaptic membrane more excitable and more responsive to the neurotransmitters that come in. So we see here that there's more AMPA receptors in response to long-term potentiation through these processes. And apart from producing just more AMPA receptors, you also see that calcium is going to go ahead and activate different pathways that are going to activate the production of that are going to activate they're going to activate the production of proteins, like they're going to activate proteins that are going to cause transcription they're going to cause genes to be transcribed and produce different proteins that are going to lead to structural changes in the cell, such as increasing the area of dendritic spines. So that is a very permanent structural change that is very different from just upregulating AMPA receptors, because these will go away in time, but if you increase the area of the dendritic spines, those are going to stay for a very long time. And that's what makes your memory stick around for a long time. And this induction of long-term potentiation will also produce signals such as nitric oxide, arachidonic acid, and others that will cause more neurotransmitters to be released from the presynaptic neuron. So the increase of neurotransmitters and also the increase of receptors and the excitability of this membrane is just going to it's like a positive feedback mechanism that increases the excitability of this one circuit in your brain. And it makes that memory stronger and stronger, so you remember it for a really long time. And that's what forms a long-term memory. This process also has inhibitors such as cyclic AMP, which will like repress this process from happening, so that they exist. The process of forming memories is really complicated, but that's kind of how it works. Do you guys have any questions about that? Okay. Well, if you guys don't have any questions, then that is it for today. You can totally contact us through GroupMe or email if you have any questions for the exam. What's up? Aaron, did you have a question? Which one? Like the first, like these two slides? $13. This one? Okay. So what happened was before we were only activating the AMPA receptors, like the green ones, because the voltage of this postsynaptic membrane wasn't high enough. But after we've activated enough of the AMPA receptor and after enough sodium has gone in and raised the potential of the cell, oh, sorry. Okay. So we have AMPA receptors and NMDA receptors. AMPA receptors were activated, sodium was let in, and the voltage increased because sodium ions were let in. And once the voltage reached a certain level, the magnesium ion on this voltage gated NMDA receptor, the ion left or was released, and calcium was allowed into the cell, right? So when calcium ions enter the neuron, they are going to activate a number of different pathways within the cell. Wait, sorry. They're going to activate a number of different pathways in the cell that contribute to long-term potentiation. One action is that it's going to cause existing AMPA receptors that were already in the cell to move up to the membrane. So the cell kind of it's always producing different proteins. It also will produce different ion channels and keep it for release when they're needed. So the influx of calcium will cause those receptors that were lying in wait to kind of go and moved to the membrane so that they can also receive glutamate and let in more sodium and further increase the postsynaptic potential of the cell. More of the AMPA receptors are produced. So it's going to cause, remember, we see that calcium activates a pathway that involves CREB, which is a protein that is involved with gene regulation. So it's going to go and cause gene transcription and the production of different receptors themselves, more AMPA receptors, to go up and also be part of this membrane and further. So that would also further increase, because the more receptors you have, the more sodium you're going to let into the cell, and the further you're going to increase the postsynaptic potential of this. And increased conductance of the ions of sodium and calcium, because the more receptors you have. So conductance is just the amount of sodium that can come in at the same time. So the more receptors you have, the more conductance you're going to have for those ions. And also, if your voltage of this postsynaptic cell is higher, then more NMDA receptors are going to be open and more calcium is going to come in, which will also increase the voltage of this postsynaptic cell. Did I answer your question? Or what else did you want to know about? Okay, cool. Okay. Well, if you guys don't have any more questions, then Arthur is going to post this recording. And you can always ask us questions over email or GroupMe. Okay. Cool.